This video is going to offer an overview of some of the applications of modern biotechnology, focusing on transgenic organisms or GMOs, genetically modified organisms, and CRISPR, a number of assays, and others. So first off, in the previous video, I introduced the idea that there are these enzymes which cut very specific sequences of DNA known as restriction endonucleases. So let's say we use such an endonuclease to cut this bacterial chromosome and make very specific fragments with what are, we sometimes call sticky ends. So these two adenines would love to bind to, these two thymines would love to bind to an adenine, these two adenines would love to bind to a thymine because we'd separated them from their complementary pairs. If we were to cut a human gene using the same restriction endonucleases, we would create the same sticky ends. And therefore, it would be rather simple then to then mix these two with the idea that now human DNA could be inserted into this bacterial chromosome. All right, so this could be done with human DNA, this could be done with any DNA. You see what a relatively simple process potentially it is. And this would then create transgenic bacteria, genetically modified bacteria that could, depending on the gene which was inserted, now make a human protein, a human product. Why would you want to do that? Well, because there are things that humans need. Humans need insulin to treat diabetics. Humans need growth hormone to treat children which have a genetic mutation where they don't make growth hormone. Humans need lots of things which would help with the treatment of cystic fibrosis or cancer or asthma, etc. And then these bacteria then would make it. Now, you don't need necessarily these bacteria. You could get insulin from other sources, like you could get it from a slaughterhouse and get animal insulin. But it isn't the same protein as human insulin, so it's less likely to be effective, more likely to cause a reaction in the patient, more expensive. So here is a product now which is better for the patient and cheaper to make. You don't need these bacteria to get human growth hormone to treat a child who needs it. You could extract the pituitary glands of cadavers, but this would be expensive. This would limit the number of children who could receive the treatment, etc. And so there are things that one can get from adding genes to bacteria. You can add genes to plants. Take a gene from one plant and put it in another plant. That could make a plant resistant to herbicides. So if there are now weeds growing in the area, you could apply a herbicide that would kill the weeds, but not kill the plant. And so actually, and I think I messed up that video. I think the herbicide resistant plant died, but you get the idea. So you could do lots of things with plants, as I'll pursue further. The first plants approved for human consumption, which were genetically modified, were those in 1994, which made tomatoes which softened more slowly so that they would ripen on the vine. So instead of adding a gene, actually one was taken away, an enzyme which would cause the tomato to soften and rot faster. So that way the tomatoes didn't have to be picked when they were green. They could then be left to ripen naturally on the plant. One can add genes to animals for lots of reasons. Maybe this is an animal which is bound for human consumption. And so, perhaps there is an attempt to have it grow faster or produce less fat and thus be healthier, etc. There have been lots of genetically modified livestock animals. Another reason to potentially modify livestock is not necessarily because one intends on eating them, but because they can then put a molecule in milk that humans would like to extract. Say there's something that you want to give patients in a human hospital. Well, you need to make that molecule. Well, if you can put the gene into an animal that would then produce it in milk, you can then simply have a herd of these animals. You milk the animals, you remove the molecule if needed, and now you can provide hospitals with something that they, they need to treat patients. You can then even treat humans by trying to, say, infect human cells with a viral particle, but the viral particle doesn't contain a virus, but rather a gene which perhaps this human patient can't make. They have perhaps a mutation and they have a genetic disease. So this therefore would be a form of gene therapy. Or if you extracted some of, say, their bone marrow cells and then infected those cells with the good gene, then perhaps this would once again be as effective as a treatment. So I'd like to, just open up with, say, like, look at all of the potential applications. Now, obviously, in real life, it's more complicated for a bunch of reasons. So, for example, just with the case of the humans. So does it work? It absolutely does. Does it cure diseases? Well, that's a little tougher. It certainly helps with diseases. So there are examples where if you take gene therapy plus the traditional therapy and you put them together, you do both, that this has a far better outcome than either by themselves. The problem with gene therapy, why it might not cause a cure, is that for some diseases, if you have mutant cells all over your body, how do you infect enough cells? So you can infect some cells, but how do you infect enough? And even if you give these cells a new gene, how do you make them express it? So they could get a new gene, but they might not express it at high levels to make the functioning protein. So it's more complicated. And then also it's complicated because obviously, individuals might vary in how they feel about a certain thing. So something I had said in the previous video on biotechnology when I teach, the way that I introduce this is I use the example of a hammer. And I ask, is a hammer right or wrong? What is the morality of a hammer? And the answer is it isn't. It's neither. It's not right or wrong. It's not moral or amoral. It's a tool. What matters is how you use the tool. If you put on a roof, that's good. If you hit someone in the head with the hammer, that's bad. And so we as a society, we make rules. We can use the hammer for this reason. We can't use the hammer for that reason. And, the society has to decide. Maybe one society would decide, differently than another society. When it comes to these applications of biotechnology, which should we use? Which precautions should we have? Which should we potentially ban? Well, the society decides. There's no automatically right answer. None of these are automatically good nor bad. The society decides. And I tell my students, you are part of society, and you, having taken a college course in genetics, you're better educated than most individuals on this. And so not only should your opinion be heard, I would argue your opinion needs to be heard more strongly. And so one of the activities that I do is I introduce a whole number of potential uses of biotechnology. And I ask my students, all right, so if you were sitting on a panel as to whether to approve this or not, should we give thumbs up and this should go forward, thumbs down, it should be stopped, something in the middle where it can stop, but with these precautions, these regulations, these conditions, et cetera. And then the lab tables discuss, and once again, there's no right answer. So while, I certainly encourage all of my students to say, like, this is how I think about this. I think this is good. I think this is bad. Once again, there's no right answer, and certainly you can have a different opinion than someone else, and many people change opinions, over time. One thing I'd like to just address before I give specific examples, one of the things that I often hear, and I try not to impose, I certainly don't want my students to have my opinions, but one thing I do try to state at some point, because this is perhaps an opinion of mine, I don't like the argument where, we shouldn't change, say, crop plants and, make corn, which is, sweeter or grows faster or whatever, because that's playing God. We shouldn't do that. We should only have natural foods, not these genetically modified foods. Okay. Now, I'm going to say something, and you're free to disagree with me. I'm not trying to convince you. This is something I find helpful in the argument. I ask my students, all right, if that's your argument, go to a supermarket and then you buy natural foods. But you tell me which are the natural foods. All right. So if you're buying broccoli, really? Broccoli is natural? You can find wild broccoli somewhere. You can find wild corn somewhere, wild wheat, all right, somewhere. There's, a natural habitat of this wild plant. There's a place where you can find wild chickens and wild pigs, not boars, not peccaries, but pigs, or wild cows, not bison or not oryx, I mean cows. And my point is, your supermarket doesn't have much natural stuff in it. Humans have been changing life for thousands of years. They have been preferentially planting seeds or crossing this strain to that one, or encouraging, these animals, and not others, because these animals gave more milk, or these animals grew bigger. These animals were hardier. These racehorses were faster. These dogs had, less of the aggressive tendencies of wolves. These apples were bigger or sweeter, etc. So we have changed life. All right, so the things we find in the supermarket, the plants and the animals, they're different than their wild cousins because humans changed them. Now, these changes occurred very gradually over long periods of time. Now, in the 21st century, modern scientific techniques allow you to make changes far more quickly. And so, maybe, that's inappropriate, but the idea that, this is fundamentally different from what has always occurred. So my humble opinion, that's just false. We have always modified nature to suit our needs better. I would like these apples better if they were sweeter. I would like this plant better if it could grow in this climate where I could plant it here where my farm is. I would like to cross this strain to this strain and see what I get. Oh, and I'll market it because it has some some attribute. So we have always changed nature. Our supermarkets are full of evidence of this. A lot of the seedless varieties, they're chromosomally abnormal. That's why they can't make seeds. And so as we then take the idea of moving genes from one organism to another, there's all of these possible applications. So once again, these individuals are talking with silly accents just so my students could distinguish between individual 1 and 2 and 3. Then I then ask them, what's your opinion? And once again, this is, I'll let you watch these videos in the playlist if you want to. I'm just going to summarize some of the points. But there's so many different applications that many students find themselves agreeing with some and disagreeing with others, and that's fine. Once again, if you were on the board which was approving this for planting or sale or human consumption or whatever, you could have an absolute position. Yes, we should always do this. No, we should never do this. But I think most people would find a more nuanced, where this one I favor and this one I don't. One of the things, once again, that plants are good at is they make stuff. All right, they make plant stuff, and very often we harvest plants, not just for food, but for like this dye molecule or this chemical which we can isolate from plants. Well, if your industry wants something, right, maybe it's something for human consumption, like a vitamin. Maybe it's just something that you want, for an industrial process, like a dye. Well, we could engineer plants that make that molecule that you want, essentially using this living organism in all its complexity as a biofactory. We could, if we want certain plants to grow in areas, once again, where we try to feed people. Once I'll revisit this conversation from time to time. Some of my students might say, no, I don't want to eat anything that's been genetically modified. That's fine. And once again, I have this little opinion. I don't insert many. But I served in the Peace Corps. I lived in a country where people were not quite as affluent as they are in the United States. And there are places where people struggle to a far greater degree. The number I'm about to say, I'm sure it goes up and down. The last number that I saw was a UN estimate that about 40,000 people will die every single day of either a hunger or a hunger-related disease. Now, you may not know any, all right? You may live in an affluent country. And so therefore, if you say, I don't want to eat anything that's a GMO, fine, all right? But some people don't have that option, that, they want to grow crops in an area that's dry and not much grows there, or it's near the ocean and the soil is salty, or something that they, would benefit from eating would suffer from frost. Well, for those individuals, frost resistance, drought resistance, salt resistance, this could make, a difference in the health of people in that area. So once again, even, as we're deciding good or bad, you might ultimately think, I personally would not eat this or do this, but I can see a scenario where some people would like to have, these crops that they could grow in areas that otherwise would not support them. One is herbicide resistance. So let's say that you don't need genetically modified organisms to apply herbicide. So what you could do is you could just fly over in a plane and dump it. You could spray it on everything. And you could try to develop a strain that after I spray it on all the crops, it kills most of them, but there's natural variations. And so here we have a strain that didn't die. It must be herbicide resistant. So I'll plant that one. Okay, so there you have a herbicide resistant plant that you didn't need biotechnology to do. You just used a lot of herbicide and you selected for herbicide resistance. So that's already part of typical agriculture. But another thing that you could do is take a gene for herbicide resistant and now insert it into this, this plant. And you could now spray the area and the weeds would die and your crops would grow. I remember reading this one story where there was this, one part of the world where there was a very tenacious weed that really limited agricultural output and how much crop plant could be grown, and so that this was helpful there. Insects plague a lot of plants, so we could spray insecticides on all of this, or you could make plants that make their own. Now, this is interesting because potentially this would, if you don't like pesticide, this could actually mean that there's less. So if you have two options, a traditional farm where you fly overhead in a plane, and you dump pesticide, or you spray pesticide over everything, or you have plants that when they're attacked by insects, they make their own pesticide there. The second scenario releases far less pesticide into the environment, where it might, contaminate streams and fish and food chains and the like. And so I know that you could make, oh, I'm an environmentalist, I'm against, GMOs for this reason. But you could also then make the argument, I'm an environmentalist, I'm for this particular GMO for this reason. It lessens the amount of pesticide which would be in the environment. And so this is not hypothetical. There are hundreds of millions of acres throughout the world which use it. So for example, if you're in the Midwest, like Kansas here, notice it's very, very dry. So there's a lot of, the dry area. But now you look here, oh, look at all the crops, and we'll see like a lot of corn presently. What's the difference? There are aquifers like the Ogallala aquifer. And so you could have some farms which are tapping into that very precious resource. But because we're using it so much, the water levels keep dropping. And so there are crops that you might like to grow in a certain area and you can't because it's too arid. Or if you did, it would cause the aquifer levels to drop more and more each year. But you could then get a genetically modified plant, which would need potentially less water. And so there is a type of corn known as drylands corn, which is being planted in Kansas, which otherwise wouldn't be able to support corn unless you irrigated it a great deal. And there may be like restrictions on how much water you can withdraw out of the aquifer. And so here's just an example. A crop that otherwise would not be able to grow in this area can, because it is a genetically modified crop. And that's just an explanation of why drylands corn doesn't use as much water. So once again, in my classes, I just then throw this out for discussion. I, certainly don't want to change your mind. I'm not advocating a particular position. You are free to have whatever position you want. I'm just, in my classes, this then fosters debate. So then here's an idea for plants, the idea of a nutraceutical. There are some foods which are healthier than others. There are some molecules which, helps the human body, or might be a vitamin that the body needs. Well, you could actually engineer plants which are healthier, right? So some, some ideas to engineer plants mean that, you don't need herbicide, or that they can grow in a new area. But some plants, you just might say, here's a nutrient that would make something healthier. Let's add it so that the genetically modified plant is actually healthier than something else. So, for example, there are fatty acids which come from fish, which are essential fatty acids needed for brain development and developing children and fetuses. And one of the things I take a supplement for these because I'm a vegetarian. And so as a vegetarian, something that the body needs that, is in a good quantity in fish. Now, obviously, I'm challenged to get it unless I get a supplement. And if you were a vegetarian, like in a country where perhaps, the vitamins, which, contain the fish oils, that's an expense that you don't want. So you might then have a choice, malnourishment, or, I can't afford this supplement. But if you could then just put engineer plants which make that essential molecule, now you could get more nutrient-rich food as a result. There are parts of the world where vitamin A is in short supply, and a lot of people develop night blindness and then full blindness. But then there are strains of plants like golden rice, which then include vitamin A. So the genetically modified plant is healthier than the traditional one, and gives a nutrient which is needed. So there's plenty of examples for these. Now, just once again, I don't give my opinions, often, but here's one. So is it good or bad? Well, if I were on a board considering this, I would want to hear the details. How is it tested? Because these genetically modified plants, very often they do undergo far more testing than a traditional strain. So let's say I'm a traditional farmer, and I say, what happens if I cross strain A to cross strain B? I don't think that's ever been done before. Look at what I get. It produces this fruit. That's interesting. I'm going to sell it at the local farm stand and say, oh, I crossed plant A to plant B. What testing do I have to do? Well, typically none. All right. Now, when you cross two different strains, those plants, those fruits, they're going to have new molecules potentially that you didn't expect. What might that, what effects might that have? Might someone be allergic? So, I mean, it is possible to have negative consequences of crosses in traditional farming. And these types of crosses, they might not require any testing to see, are we sure that, no one would develop an allergy or to this or whatever. These genetically modified plants, they are tested. There are rules and regulations as there should be. Are there sometimes problems? Yes, there are. So there have been a handful of examples where something wasn't yet approved for human use, but it made it into, human products. There were no ill effects in the example that I'm thinking of, but nevertheless, that needs to be addressed. That doesn't necessarily, directly affect. It's just more the protocols. How do we enforce the protocols? Some people might say, well, I don't want, like your strain of genetically modified stuff producing pollen that blows over into to my field and, crosses with my plants, that's a good concern. And that, once again, that's not necessarily the, logistics of that, but then how is it implemented? Do things have to be spaced a certain way, et cetera? But once again, if I was a traditional farmer and I developed some new strain, there's nothing stopping the pollen from my plants from ending up, fertilizing the flowers of your plants as well. So in any case, there's lots of potential applications of green biotechnology. I'll get to, I think, a few more when it comes to energy in just a little bit. And so I asked my students, which would you would you favor if you had that ability or not? Also, so I'm using green biotechnology, blue biotechnology, because there are just so many applications. Some people have said, well, if we split them into these categories, then that lets us, organize our thoughts better about, what we feel about this category versus that category. In blue biotechnology, it's biotechnology affecting aquatic organisms. Now, I know we want to think about fish first, but a big one is algae. All right, so I mean, algae are living in water, doing photosynthesis, and they're making stuff. Could we make them into factories? And one of the things which is receiving an awful lot of research is the idea of, what do we need? Well, we certainly need fuel, all right? And things like hydrogen gas can be produced by algae and can be used as a fuel. When we think of what petroleum contains, it's just carbon hydrogen chains. Could we engineer organisms which make some of the byproducts of petroleum processing? Biodiesel. So could we get the same energy sources, but not from the remains of living things from long ago, but from living things which are making them today? So there are certainly lots of applications of this. One I used in my previous video. The thing that makes PCR so advantageous is that there was a DNA polymerase isolated from bacteria, aquatic ones, which live at very high temperatures. And so therefore their proteins can tolerate high temperatures. So that's why you can have this enzyme in the PCR reaction that you have to take to a very high temperature. So we get a lot of useful products, antibiotics, enzymes, and the like from aquatic organisms. So that's one aspect of blue biotechnology. Another aspect is potentially we could change organisms which we intend on eating. All right, and there was a type of salmon, for example, which was genetically modified, which would then increase, which would grow to adult size in half the time. Now, we could once again debate this, and if you were in my class, I would expect you to debate this with your lab partners. Well, have we controlled for this? Have we controlled that it's not going to fertilize wild salmon and then change wild populations, et cetera? How do we control? So it should be studied. It has, and there are no adverse, health or environmental effects to date. Now, once again, your decision on whether you favor this or whether you think it should be done, that's completely up to you. The society decides what's right and what's wrong, and you as an individual can decide what you buy at the grocery store, et cetera. Once again, I just sometimes make the point, even if this isn't something for you, even if you're thinking, I would never ingest this, there are an incredible number of people who need more protein than they get. All right, so protein sources are often in short supply. Just, for example, Southeast Asia certainly depends a lot on fish and aquatic life, but it's also the most densely populated part of the world. And a lot of their fisheries, have been overfished so that the fish stocks are declining or crashing. So how do you ensure that people get enough protein? So even if you, who doesn't have to worry about having access to protein sources, isn't in favor of a protein source which can grow to adult size in half the time. That doesn't mean that it isn't something which wouldn't potentially uh, benefit other individuals. And lots of people are studying aquatic organisms once again to realize, oh, this agent in sharks has an anti-cancer property, or this thing from marine fungi is an antibiotic. And so once again, some of this biotechnology is just trying to isolate useful molecules, which we can use for lots of things. Some people refer to things like white biotechnology or gray biotechnology as, gray potentially helping, the environment. So you're genetically modifying organisms to make the environment better, or white biotechnology where you're just trying to generate, chemicals. And so once again, here there are some examples if you wanted to consider these. So one of the early ones is, there are things about the environment we would like to change. Like when we spill oil, we would like it to get cleaned up. If we pollute soil with heavy metals or acid mine drainage from a strip mine from coaling or coal or whatever, or get mercury out from the coal, we could make the environments better. Well, could we engineer microbes to break down petroleum so that you could release them at an area of a petroleum spill, and then it would limit then the effects of this oil on wildlife, et cetera? Could you then get microbes or plants which suck up which use the heavy metals, which remove it from the soil, in which case the environment is getting better because of the use of these genetically modified organisms. Another thing is fuel. All right, so you can, we can get ethanol and add it to gas. So we're using less petroleum because we add ethanol to it. So this could be the residues of, of corn and crop plants. But you could grow trees just with the idea that, it's going to generate ethanol. So you could genetically modify plants so that they grow more quickly, or that they are more efficient in the production of these biofuels. One of the negative things is now that ethanol from plants can be added to fuel. In some areas like corn, you have an issue. All right, people are growing corn, but the more corn you're growing for fuel, often for richer countries, then that's the less corn you have for food, often for poorer countries. And so now you could specifically have corn engineered not for food, but for fuel. And so that there's no, you're not, taking from the food corn to make fuel. Once again, there's the hope that, petroleum, it's just hydrocarbons that originally came from living things. And then, there was pressure and heat that changed it. But could we generate, could we engineer organisms to make this? Because if you could, then the same thing you could get out of petroleum, which would be making climate change worse because you're taking petroleum and carbon molecules which were not in the carbon cycle and adding it to the carbon cycle. If instead you grew plants which take carbon out of the air into making something like a petroleum that could be used in as a fuel, then you're using petroleum, I don't know, run a car or something, but it's not adding carbon to the atmosphere because it's not taking carbon which was sequestered outside of the carbon cycle and putting it into it. It's just taking out of the carbon cycle and then putting back in. And so this would then be a renewable form of a carbon. So there are so many applications. And once again, some of those videos, the goal was, if you had a chance to say, I approve of this, I don't approve of this, what would it be? There's no right answer, but once again, societies, and then you compose society, your voice should be heard. And hopefully then, the better informed your voice is, obviously, the better. So what is your opinion? So this invites you to explore. Now, some of the issues coming out of this are not biological issues necessarily. So for example, should we label genetically modified plants? Well, some of the arguments then are, well, if we labeled them, then say, here's a common argument, then European farmers and European populations in general tend to favor GMOs less might be now less inclined to buy American exports. And so some of the reasons really aren't biological or health oriented, but now more economic, where we want to, this country wants to export as much as it can. And if this was enacted, then that would limit exports. So there's lots of issues, and once again, classes would be free to discuss it, but that doesn't quite get into the the value of this per se. One technique which is related to this is what's known as CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now, CRISPR originated in bacteria, and it is a defense that bacteria use to learn how to better fight viruses in the future. Now that's just impressive because we vertebrates and we higher vertebrates, we think very highly of our adaptive immune response, which can learn and get better over time. Like insects don't have much in this. But here's a bacterium, which essentially has an adaptive immune response where it can get better at fighting viruses over time. So just let it be said that, the fact that bacteria have an adaptive immunity, that's impressive. The steps of how bacteria use it is that in their first step, they cut the foreign DNA, say from a virus, and they attach it to specific proteins. So they're recognizing, here's a problem. We need to be able to recognize this in the future. And then these proteins then insert this into the host chromosome. So they're taking foreign DNA and inserting into the host DNA, but at a specific site between palindrome sequences. And so there's these base pair repeats. Palindrome is something that reads the same way in the forward and backwards direction. And so this foreign DNA is now part of the host chromosome between these palindromes. All right. So step one was to get the foreign DNA at this spot. Okay. Then step two is expression. You then express the foreign DNA and make it into an RNA. All right. So from the DNA sequence, an RNA copy is made. And so this is known as the precursor CRISPR RNA. And this will then bind a protein. So now you're going to have what's called a ribonucleoprotein that has an RNA component and a protein component. So there's CRISPR RNA plus associated proteins. It forms a ribonucleoprotein. But since this is the sequence of foreign DNA, then the final step that the bacteria use it for, the interference step, is that this can then seek out and destroy, or at least interfere with, any more foreign DNA which has that sequence. So you've identified this is something to look out for. This RNA then guides these proteins to foreign DNA, say from a virus, and allows the bacteria then to eliminate the threat from this virus. So that is how bacteria use CRISPR. But see, the beauty is it can take DNA and insert it into a specific site. And so in biotechnology, there is great interest in this as far as the ability to guide new DNA and insert it into a new spot. So the applications of CRISPR are potentially enormous, as would be then the need for regulation. CRISPR can be done, here, but not here in these cases, et cetera. There are so many other applications of biotechnology, and I have more videos, but I just want to introduce those with the idea that you could then seek them out if you saw fit. But then also just when I teach, I'll just be honest, we run out of time. All right, and so another big application of this would be the use of antibodies and other probes to find things. All right, so that's the beauty about, a lot of times you want to find something, And a lot of times just in a purely scientific sense, you want to ask, what proteins are being made in the cell? Where, I know humans have this gene. What cells in the body use that gene? Where is it? And so if you can have things that stick to the molecule you're looking for, you can find it. And you can say, oh, liver cells make this gene, or, oh, this cancer cell is making this protein that normal cells don't make, or whatever. So there can be antibodies. There are differences between monoclonal antibodies, polyclonal antibodies. I go through these. Once you have antibodies which stick to molecules, you can do a diverse number of assays because the antibody is sticking to the thing that you want, and there might be a scenario where, it will form like a solid precipitate that you can actually visualize. So if antibody can stick to the matching molecule, what's known as the precipitin test, you can actually see it. So patient number 1 is making this thing that the antibodies bind to, but patient number 2 is not. And so there are so many assays. And so, for example, if you were to do, a test for HIV or a test for Lyme, I mean, I often get Lyme's tests. I've had Lyme a couple of times. And so you are going to use an ELISA test. And so, that involves antibodies and sometimes secondary antibodies. Once again, I have videos on this on different types of immuno assays, and I, invite you to look at the ones that, apply to you. And one of the things, like if you can find things, you can say, oh, like this patient is making antibodies against Lyme. They must have been exposed to them. This person is making antibodies opposed to coronavirus. They must have been exposed to it. Or look, the probe for this against this type of protein is binding to the cancer cell in a different way than a normal cell. This protein seems to contribute to cancer when abnormally expressed. Or just if we're trying to figure out what a protein does, all right, like, oh, this protein, it turns out, is expressed in, it functions in the nucleus. This protein exists only in mitochondria, etc. So some probes are radioactive. Some are fluorescent. Some can change color when you add the appropriate reagent. And so once again, probes just allow you to find things and visualize things. So there's, one of the aspects of biotechnology is this great variety of probes and assays which let you test for certain things. And so once again, the better we understand the DNA, RNA, and proteins, and the functioning of life, the better we can, manipulate these as tools. And so biotechnology certainly has an exploding set of applications, but not every application is necessarily appropriate or ethical. And so it is the role then of society to then decide which of these applications to allow and under what conditions. And I hope, that you have opinions on this and that, you can be active in, the, informed conversation that our society has.